Hello and good morning, everybody. Welcome to GemPharmatech's monthly webinar series. My name is John Jackson. I'm part of the marketing team here. And today you'll be hearing from Dr. Mark Johnson, our Senior Director of Business Development, as he covers the topic from off-the-shelf to purpose-built, optimizing mouse models for mechanistic and translational research. Before we get started on that, I'll take a few minutes to briefly explain why or who GemPharmatech is. Our mission is to engineer a healthier world by pushing the boundaries of biomedical innovation. The way we do that is by providing the models, data, and insights to move drug discovery forward. And specific to the models, we have the largest collection of genetically engineered mouse models in the world. This includes our knockout collection, which Mark will be covering today, as he also covers custom model generation. And especially for our biotech and pharmaceutical clients, we specialize in preclinical efficacy testing services, and we do this both in our headquarters and facilities in China, as well as our facility in San Diego, California, which we're happy to announce recently was ALAC accredited. If you have any questions that fall outside of the topic of discussion today, feel free to contact us at sales@gempharmatech.com. At the end of this webinar, there will be a Q&A session, and we will use the Q&A function on Zoom to receive your questions. So if you have any questions, that's how you will communicate them. And please follow us on LinkedIn, and where you can find the next month's topic for the webinar and where you'll be able to find this on demand. With that, I'll leave it to Mark for him to get started on this topic.
Thanks for that introduction, John. Good morning, everyone, or good afternoon, depending on where you are. Thank you for joining me today. In this session, I'd like to talk about model selection, specifically how to align genetically engineered mouse model strategy with the stage and goals of your research program. We're in a remarkable era in vivo research. The scale of available models has never been greater. Genome-wide knockout resources exist. Humanized strains are increasingly sophisticated. Custom engineering is faster and more flexible than ever, and yet the abundance of options has made decision-making more complex. So rather than simply reviewing model types, today I'd like to focus on strategy. How do you choose the right model for the right stage? And how do you avoid decisions that may introduce downstream risk? Model selection is one of the most consequential decisions in early in vivo research. It determines biological interpretability, it shapes translational relevance, it influences time to data, and it affects downstream development risk. If the model is misaligned with the biological question or therapeutic modality, you may generate clean, reproducible data, but data that doesn't translate. And often that misalignment isn't obvious until much later in development. In other words, model selection is not just a technical choice, it's a strategic inflection point. So before we discuss specific engineering strategies, I'd like to zoom out and look at how model strategy aligns with stages of drug development. What this slide illustrates is that model needs evolve as a program matures. In early discovery, the goal is broad pathway interrogation. Conditional knockouts are powerful here. As we move into target validation, we may need conditional strategies, particularly if lethality or systematic effects emerge. During mechanistic studies, spatial and temporal control become important. Inducible and tissue-specific systems help refine our understanding. In preclinical development, alignment with therapeutic modality becomes critical. Point mutation knock-ins and humanized alleles allow us to evaluate pharmacological engagement and patient-relevant biology. And by the time we reach IND-enabling work, the model must closely mirror the pharmacology and translational context expected in humans. The key message here is simple. Model strategy should evolve alongside your program. And recognizing when to leverage off-the-shelf resources versus when to pursue custom engineering is central to that evolution. But how do you actually decide what to build or what to source? I find it helpful to think about model selection across three categories. First, the genetic and molecular design. Are we talking about a constitutive knockout, a conditional knockout, or a knock-in? Does the strategy preserve endogenous expression? Is there a specific human mutation that needs to be modeled? And importantly, are there compensatory pathways that may mask phenotype? At this level, you're defining the molecular precision of the model. Second, biological context. Does the gene need to be manipulated in a specific tissue? Is temporal control required? Is there risk of developmental lethality that would necessitate conditional control? And how does strain background influence phenotype? These considerations often determine whether a straightforward strategy is sufficient or whether complexity is required. And third, and this is often underappreciated, translational and program factors. What stage of development is the program in? Does the model need to support drug target interaction studies? Is there an existing validated strain available? And what are the timeline constraints? Sometimes the scientifically ideal model is not the strategically appropriate one. So model selection sits at the intersection of molecular design, biological context, and program strategy. When these three dimensions are aligned, the model becomes an accelerator. When they're misaligned, it becomes a bottleneck. And with that framework in mind, I'd like to first look at the landscape of available resources. What this slide illustrates is the scale of genetically engineered mouse model resources worldwide. Across major repositories, including Jackson Laboratory, MMRRC, Riken, EMMA, Taconic, and others, tens of thousands of engineered strains are available globally. This ecosystem has dramatically expanded access to knockout, conditional knockout, reporter, and disease-specific models. It's an extraordinary resource for the scientific community. But abundance alone doesn't solve the decision problem. The real question becomes, which repository offers the depth, validation, and engineering flexibility aligned with your program? According to IMSR data as of May 1st of last year, GemPharmatech maintains one of the largest and most comprehensive gem collections worldwide, with over 22,000 strains represented. This positions GemPharmatech among the global leaders in scale and coverage. The scale alone is not the differentiator. What matters is how these strains were built, validated, and supported. The Knockout All project, or KOAP, was built by GemPharmatech with a very specific intention. The goal was not simply to generate individual knockout strains. The goal was to systematically engineer the mouse genome at scale in a way that supports both constitutive and conditional biology across the majority of protein coding genes. This was a long-term infrastructure initiative. Rather than developing strains reactively gene by gene, KOAP was designed to create a standardized genome scale platform. Historically, many knockout models were generated in academic settings, often with unique targeting strategies, varying genetic backgrounds, and inconsistent validation standards. KOAP was built to address that fragmentation. The intention was genome coverage, standardized allele design, conditional ready architecture, and reproducibility. Today, KOAP includes over 22,000 engineered mouse lines, including more than 11,000 constitutive knockouts and over 10,000 flocked alleles. The level of coverage allows rapid access to a broad range of targets, often without initiating full custom model development. Importantly, KOAP is not limited to conventional knockouts. The collection includes conditional ready designs, tool strains such as Cree lines, reporter strains, and an expanding portfolio of humanized disease alleles. This provides flexibility, particularly as programs evolve from pathway interrogation to translational alignment. One of the strengths of KOAP is standardization. Most strains are built on a C57BL6 background with consistent targeting strategies. This reduces variability and simplifies downstream breeding and interpretation. And disease-relevant strains undergo ongoing validation. So what does that mean in practical terms? KOAP enables rapid target interrogation without initiating full custom engineering. For early discovery and target validation, this can significantly compress timeline and reduce risk. The KOAP collection is particularly powerful in rare disease research. Because many rare diseases are driven by single gene defects, genome scale knockout and conditional ready resources provide immediate starting points for disease modeling. Instead of initiating full custom development for every new gene, researchers can often leverage existing KOAP strains to rapidly interrogate gene function, phenotype relevance, and therapeutic hypotheses. Across neurological, cardiovascular, metabolic, renal, hematological, and other rare disease categories, KOAP offers broad genetic coverage that accelerates early target validation and mechanistic insight. In rare disease programs where time, funding, and patient populations are limited, that ability to move quickly from gene to in vivo data can be especially impactful. This slide illustrates how KOAP-derived knockout strains can function as a rare disease model. ILMS1 encodes a centrosomal protein involved in ciliary regulation. In humans, mutations in ALMS1 cause Alström syndrome, a rare multisystem disorder characterized by childhood obesity, type 2 diabetes, cardiomyopathy, renal dysfunction, and vision loss. The C57BL6 ALMS1 knockout strain from KOAP collection recapitulates key metabolic phenotypes observed in patients, including progressive obesity, hyperglycemia, and elevated insulin levels. That's important here, not just that the gene is knocked out, but that the phenotype aligns with human disease manifestations. This is an example of how a genome scale knockout resource can provide immediate entry into rare disease modeling, enabling rapid mechanistic studies and therapeutic hypothesis testing without initiating full custom development. One way to assess the robustness of a large-scale resource is to look at its scientific output. To date, KOAP-derived strains have supported over 1,100 peer-reviewed publications with a cumulative impact factor exceeding 13,000. These studies span top-tier journals including Nature, Science, Nature Biomedical Engineering, and others, and cover diverse biological systems from metabolism and immunology to neurobiology. This publication record reflects not just scale, but reproducibility and scientific utility. It demonstrates that KOAP strains are not only available, but they are actively driving high impact discovery. This is a very important slide. It answers a practical question. When should you move from an existing model to custom model generation? And the answer is if the existing models that are available don't meet one or more of the criteria that I've listed here. From a scientific standpoint, the most important question is this: does the model truly reflect the biology your therapeutic is trying to modulate? That means preserving endogenous regulation when necessary, avoiding developmental lethality if it confounds interpretation, matching loss of function versus gain of function to your therapeutic mechanism, and increasingly modeling the actual human genetic variant that is central to your program. If the available models fail on those dimensions, that's a strong signal that custom design may be warranted. Then there's the programmatic layer. Even a scientifically elegant model may not be viable if it does not fit your developmental timeline, your budget, or your intellectual property strategy. Freedom to operate, ownership of the allele, contracting timelines, quarantine, and colony expansion, these are not secondary details. They directly impact how quickly you can generate data and protect your asset. So custom generation should not be the default, but it becomes the right choice when scientific alignment and strategy control are both critical to program success. Before we dive into specific model design strategies, it helps to step back and frame the major categories of genetic modification in rodent models. At a high level, we're typically working within 4 buckets: gene knockouts, gene knock-ins, complex modifications like humanizations, and transgenic, where we can introduce or overexpress a gene fragment through a controlled or random insertion. All of these can be built on different genetic backgrounds depending on the biological question. Today, there are 2 primary approaches used to manipulate the mouse genome. The first is ESL targeting, based on homologous recombination in embryonic stem cells, followed by blastocyst injection. This is the more traditional and highly controlled approach. The second is zygote microinjection, where DNA and/or nucleases such as CRISPR-Cas9 are injected directly into fertilized oocytes. This method is faster and more flexible. Most modern model generation strategies build on one of these 2 foundational platforms. You can see that I put slow and fast in quotation marks because these are referring to the timeline to founder generation, not necessarily the timeline to study cohorts or data. So I'm just going to briefly run through the different model generation types and some considerations that you should evaluate when choosing your model. So constitutive knockouts. This is pretty straightforward. You just need to inactivate genes. And in general, CRISPR-Cas9 is the right tool for this type of model. It's fast, it's quick, and there are many options because you can delete different sets of exons. The only thing to look out for here is if the knockout of the gene is predicted to have a developmental lethality, or if the background strain could affect the phenotype that you get in the end. Conditional knockouts, these are great for avoiding developmental lethality or to have tissue-specific or temporal-specific control of gene function. So the thing to look out for with Cree lines is that you, sorry, with Phlox lines, that you also need a Cree line, right? So as you're developing your Phlox strategy, you should already be thinking about which Cree driver you're going to use and the increased time it's going to take to cross these alleles together and do different temporal or spatial induction. I just want to point out here that while you're looking for Cree lines, that GemPharmatech, as part of the KOAP collection, has over 250 Cree strains already developed and validated. And these span multiple different therapeutic areas and tissue types. So this is a great resource to pair with either Phlox alleles in the KOAP collection or with custom generated Phlox alleles. We also have Cree dependent reporter lines, as the one demonstrated on the right here, where you can see TD tomato expression in the pancreas only in mice that have the Cree driver line. Knock-ins. So I'm just talking about knock-ins generally. This could be knock-in of a reporter, it could be a knock-in of an epitope tag, it could be a knock-in of a humanized sequence, which we'll talk about in a second. The major consideration here is that there's different ways to introduce genes, primarily one way that will disrupt the mouse gene function and one that will preserve it. So you can either replace part of the mouse gene so that it activates it, or you can use things like bicistronic sequences like T2A or iris sequences to add the insertion after the coding sequence of a gene. So this would preserve endogenous gene expression. Genetic humanization. So there are several ways that you can do genetic humanization, which is the replacement of mouse sequences with human sequences. This is primarily for translational research. So seeing if your drug will interact with the human target in a biological system that the mouse is a surrogate for. So your strategy here is really important because it depends on how your target is going to engage with either the DNA that encodes the target or the protein target itself. And how you do this depends on many factors. Just to name a few, the size of the human gene, what part of the gene that you're targeting, and some of the downstream breeding considerations. So just a few examples here, you could do a full replacement that includes the introns of a mouse gene, replacing them with the human exons and introns. You can do a mini gene, so you just knock in the coding sequence of the human protein right at the ATG of the mouse sequence. You could do a partial replacement. So this is great if your drug, let's say, only binds to the extracellular domain of a protein, but you want to keep all the intracellular signaling and mechanism the same or as mouse sequence. So you can selectively do humanization of just part of genes. So replacing just exons that encode a certain domain of a protein. And then particularly important for nucleic acid therapeutics that are targeting the DNA itself of a gene, a lot of times you need the intronic sequence or even the UTR sequence at the 5' and 3' end to be intact. So you can include in the replacement region these UTRs. Just another mention here, on top of the KOAP collection, GemPharmatech also has a pretty huge collection of humanized alleles already developed. So these are generally targeted to different disease areas. So these were intentionally built, and they are all designed specifically to interact with the predicted drugs that are available. Random integration transgenesis. So this is something I always like to talk about because many people have the misconception that making random integration transgenes is the fastest way and easiest way to make a custom model where you get overexpression of the gene of interest. So it is a great way to test different variants very quickly because you can generate founders very quickly by just inserting exogenous DNA into a fertilized egg. However, these sequences go into the genome randomly, so the location cannot be predicted. They go in different copy numbers, they go in different orientations, and these so-called transgenic arrays can be quite complex. So that could really confound how your therapeutic is engaging with the target. It could confound how the sequence is expressed in the mouse, depending on the context in which it was inserted. So it may be influenced by neighboring genes, for instance. And also, founder generation is not the end all of this project. That's just the beginning. So each founder you generate has a different transgenic insertion. So each one would have a different phenotype and a different presentation of what you're looking for. So you may need to breed several founder lines at the same time and screen all of them in order to establish the one that is working for your project. And then you would need to expand it after that. So a lot of times, random integration transgenesis can actually be the longest timeline. One way that GemPharmatech has provided an alternative option for transgenes is a targeted transgenic. So we are using CRISPR-Cas9 to insert transgenic cassettes or small pieces of backs into a safe harbor locus. So this ensures that the gene of interest goes into a location in the genome that is predictable, can be tracked, is epigenetically open for expression, so allowing the promoter to actually drive expression in the tissues that you anticipate. And it preserves a more native and natural endogenous copy number. So you either have one copy or two copies, not 10, 20, etc. And then finally, we have point mutations, which in general in drug discovery is trying to introduce a disease variant in the human population into a mouse gene and replicating that effect. This also is generally done by CRISPR-Cas9. It's a very easy and efficient process to make small single nucleotide or small sequence insertions. The only caveat to this approach is that oftentimes we have to engineer silent mutations into the insertion as well for screening purposes. So let's say, for instance, your drug was a siRNA that targets this region. That silent mutation may disrupt binding. So I thought I would just throw out one example of a translational mouse need so we can kind of go through the criteria here and some of the thought process on what model you would choose. So here's a scenario where a drug company wants to develop a therapy for a target that is a GPCR that regulates glucose metabolism. As you may have noticed, these are very in vogue at the moment, and this would be to treat diabetes and/or obesity. And their therapeutic modality is an siRNA. So for this particular target, there's no existing humanized models already in repositories. The target sequence is intronic and species specific, and the target engagement and efficacy studies must be completed within 2 years. So what other considerations should you think about here? So will the human variant compensate for mouse functionality? Does expression pattern recapitulation matter? And does gene dosage or genomic context matter? And I would say in this case, all three matter. So what are the options? As we kind of just discussed, you could use a back transgenic approach where you insert the entire gene, including the surrounding regions, into the mouse genome. The benefit would be that if there is more trans regulation of this gene, you would more likely include that by inserting a larger piece. However, this is going to go into the genome in a random place that could be affected by surrounding sequences. So that's a risk there. And then also you would probably have to screen several different founders in order to get one that is not affected by the insertion site, has a copy number that's in the scope of what you're looking for, and that does not have any confounding kind of transgenic array issues. Another approach would be the humanized knock-in. So we could, if the gene is small enough, we can just replace the mouse gene with a human gene, including the introns or UTRs that are the targets for the therapeutic. Copy number and insertion site would be controlled in this case, and the expression pattern should match what is in the mouse, not necessarily what is in the human, however. So you may lose function of the mouse sequence by replacing it in this case. That's a risk. There's limitations on, as I mentioned before, how big you can insert. So I think right now our GemPharmatech's limit, at least I think, is something like 8 kb. And then, yeah, when you make different chimeric insertions like this, it may affect the gene regulation and splicing of this insertion. So now I'd like to talk a little bit about time consideration when it comes to model development programs. One of the biggest misconceptions in model development is that speed equals founder generation. But time to founder is not the same as time to cohort readiness. As you can see here, model development is a pipeline from design and FTO analysis all the way through cohort production. Each of these steps adds time and, more importantly, risk. If you rush early design decisions, you often pay for it later through founder rescreening, poor germline transmission, unstable alleles, delayed validation, or inefficient breeding strategies. Upstream strategy determines downstream compression. If the allele is designed correctly, if validation plans are built in early, if colony expansion and IVF strategies are considered upfront, the overall program timeline can actually be shorter, even if founder generation itself takes a little longer. So the goal isn't simply speed, the goal is intelligent compression of the entire development arc. If time to cohort readiness is the true metric, then the question becomes, how do we deliberately compress this entire arc? There are four major levers. First, provider and platform selection. An experienced model generation team reduces technical risks. Clear FTO and IP frameworks prevent legal delays. And integrated validation and breeding infrastructure avoids handoffs that slow programs down. Second, strategy alignment. Model design must reflect program constraints from day one. Founder and screening strategies should be optimized for the biology, not just speed. And design and FTO analysis should run in parallel, not sequentially. Third, reproductive acceleration tools. IVF can dramatically compress colony expansion and even assist germline transmission. Cryopreservation planning early prevents future bottlenecks. And finally, integrated execution. In-house molecular validation, scalable accredited breeding, and optional preclinical services eliminate transitions between vendors, which are often hidden sources of delay. When these elements are aligned, the result is not just faster model generation, it is faster, lower risk progression to data. As we come to the close of the webinar, I'd like to return to the central theme of this talk. Model selection is not a technical afterthought. It is one of the most consequential decisions in early in vivo research. The model you choose determines biological interpretability. It shapes transitional relevance, it influences time to data, and it directly affects downstream development risk. We discussed how time is relative, that founder generation is only one piece of the program timeline. Intelligent upstream decisions determine whether your downstream phases compress or expand. We reviewed how off-the-shelf resources like KOAP can accelerate discovery, particularly in rare disease and target validation settings. And we explored when custom model generation becomes necessary, especially when scientific precision, IP control, or program alignment demand it. Ultimately, the most successful programs are not those that move fastest at the first step. They are those that are strategically aligned from design through cohort production. That's why working with an experienced partner matters. A fully integrated platform spanning genome scale resources, custom model generation, validation, scalable breeding, and preclinical services reduces fragmentation, minimizes handoffs, and lowers technical and operational risk. At GemPharmatech, our goal is to serve not simply as a vendor, but as a scientific partner with extensive model resources, therapeutic area depth, and the infrastructure to support programs from concept to in vivo data. If you're designing a new program, evaluating an existing model, or considering a custom strategy, I strongly encourage you to consult with experts early. Early alignment prevents downstream delays. Thank you for your attention, and I'll be happy to take any questions.
So now will be the Q&A portion of our webinar. If you do have a question, please type it into the Q&A module on Zoom. We have a few already, but I'll give everyone a chance to type one out of their own. It looks like there's one. I'll read it out loud. How do you decide when a simple knockout is sufficient versus when you really need a humanized allele to support translational development?
Well, a knockout versus a humanized allele are kind of like apples and oranges generally. They're usually for different purposes. So a knockout is generally to assess loss of function of a gene, prove that a target is worth pursuing. So it causes a disease or it causes a specific phenotype. A humanized allele is generally you now have a therapeutic or a drug that is targeting specifically the human protein, but you just want to evaluate it in a biological living setting. So uh, basically, when you're moving into drug development and you actually want to interrogate whether your therapeutic is engaging or having efficacy, that's when you need a replacement knocked in humanized allele.
Let's choose this one. For biotech companies thinking ahead to partnerships or IND enabling work, how early should they be thinking about FTO and ownership of their mouse models?
They should be thinking about this as soon as possible. I used to be a custom model generation subject matter expert, and I've worked with a lot of biotech companies and pharma companies to develop various custom models for their programs. And a lot of times, this subject would come up as an afterthought after they already planned which mouse they wanted to use or what design they wanted to use. Um, we would often run into hurdles like trying to recapitulate, especially in biotech or for-profit research. People trying to recapitulate things from academia, there's often different hurdles there. So specifically around IP, a lot of times the base models for things or the base backgrounds are owned by an academic lab or a different nonprofit organization. So if you find out early that it's going to take you 6 months to a year to get freedom to operate, you may want to pursue a different target or a different strategy or even a different program because that could really kill any chance of you getting the data you need within a specific timeline. I think that's kind of the overarching message that I want to impart from this webinar is that there are, the laundry list of considerations for how you should choose the model and or how you should design it. I strongly recommend that you consult an expert, whether you decide to make the model yourself or use a vendor to generate it. I would consult someone who's done this many, many times, and they can kind of like look out for these red flags.
You mentioned that founder generation isn't the same as cohort readiness. Could you expand a bit on mosaicism and founder variability and how teams should think about screening strategy early on?
I think this is often um, not thought about ahead of time. People are so focused on getting founders or getting F1 heterozygotes as fast as possible. And if you look at the various service providers, the deliverable is generally like, I don't know, 2 breeding pairs or 3 to 5 mice. And anyone who's ever worked with mice, you can't really do much with that, with those mice. That deliverable is often like very precious. If you lost, if you didn't have a male and a female, you wouldn't be able to breed them. And if you have that few number, you need to basically dedicate them to breeding to expand the colony. So you don't really have any leftover animals to do validation on. So if you think ahead of time to set your deliverable or cohort sizes to be scaled at the right size at each step, that will allow you to not churn and waste time and do things iteratively. So when it comes to like founder development and mosaicism, you should choose a strategy that is likely going to give you a high rate of mosaicism, so high rate of the gene being introduced. Or if you know that it will be a low rate, you should scale up the production of those founders. So if you predict low mosaicism because of the technique, I would double or triple the amount of founders that you're generating. And furthermore, from there, I would probably not risk things by doing natural mating. I would use an IVF to make sure I get enough animals at that end of that model generation process to do validation, to set up interbreeding, to expand the colony. That'll save you a lot of time.
If you have any questions that you didn't want to share live, feel free to reach out to us at sales@gempharmatech.com. And we look forward to welcoming you to next month's webinar. Hope everyone has a nice day and see you soon.
Thank you.